Alrighty, so hi guys, my name's Alyssa Repture, and I'm going to be doing my senior seminar presentation on how a human monoclonal antibody can block the SARS-CoV-2 infection. So this SARS-CoV-2 is basically the virus that causes COVID-19, and I became very interested in looking into the therapeutic methods that are currently being researched for COVID-19 due to the pandemic that we're currently living in and how it has had such a huge impact on all of our lives and continues to every day. And so I found this article and I was immediately interested. So, just going to start with some background surrounding COVID-19, the pandemic itself, and then go into a little bit of background on SARS-CoV-2, which, as I stated, is the virus that causes COVID-19. And then go into some background on the proposed therapeutic method from the particular article that I researched. Then I'm going to go through some of the methods that were used to discover the specific antibody known as 47011 and the properties that were discovered through experimentation, and then the results that came from these methods. And then lastly, I'm just going to go over the conclusions from the article, as well as how these results and conclusions are very promising for treatments looking forward. So just a little bit about the COVID-19 pandemic. So COVID-19, it stands for coronavirus-induced disease 2019. It is caused by SARS-CoV-2, which stands for severe acute respiratory syndrome coronavirus 2. This virus originally emerged in Wuhan, China in late 2019 and belongs to the sarbecovirus subgenus. Also belonging to the sarbecovirus subgenus is SARS-CoV, another virus that originally emerged in 2002. It did infect approximately about 8,000 people, but had nowhere near the amount of impact that SARS-CoV-2 has had on society globally. Both of these viruses are thought to be zoonotic, meaning that they have crossed a species barrier. In the case of SARS-CoV-2, there's been theories leading to SARS-CoV-2 coming from a bat and then crossing a species barrier to a human. And then a statistic that I just wanted to include from the article was, as of April 2020, was approximately 2 million cases or over 2 million cases that were reported. And we know today that that number has significantly increased. Common symptoms of coronavirus we know are fever, loss of appetite or taste, fatigue, cough, and shortness of breath. We know that patients that have the disease are either symptomatic or asymptomatic, and the main source of infection is COVID patients themselves. So, being in close contact with other people who are testing positive for COVID-19 for an extended period of time is the main source of infection. And so, just some background on the virus itself. So, this virus has a positive single stranded RNA genome. And then I also wanted to mention some of the proteins that are characteristic of the virus because they are very important in the virus replication as well as maintaining the viral genome. And those proteins are the membrane protein, the envelope protein, a nucleocapsid protein, which attaches the single stranded RNA genome to the membrane of the virus. And then one of the most important proteins in terms of treatment and vaccine design currently is the S-glycoprotein or the spike protein, which we can see here is this red spike. And so this protein has a trimeric structure. It binds to human mucosal epithelium, which lines our nose and our mouth. And it binds this epithelium via an ACE2 receptor, which is known as an angiotensin converting enzyme 2 receptor. And so when the spike protein binds to the receptor, there's a conformational change that causes membrane fusion of the virus and the host cell or the human cell, and thus causes infection in the human cell. And so I wanted to include this photo because it really shows, like visually, the proteins and where they are in terms of the virus. And so here we see the trimeric structure of the spike protein. You can see the membrane proteins here. The nucleoprotein or the nucleocapsid protein attaches the genome to the membrane. And then we see the genomic RNA here. And then lastly, we have the envelope protein here. And so the proposed therapeutic method from this article is that if we can take neutralizing monoclonal antibodies that target the surface protein, then the SARS-CoV-2 can possibly be neutralized and the infection neutralized as well. And so as we can see in this photo here, if the neutralizing antibody were to bind to the S protein, then the interaction between the ACE2 receptor and the virus can be neutralized. That is not to say that it would be compromised completely, but somewhat neutralized. And so this is basically the main target of therapeutic and vaccine design currently, because the S protein and the receptor interaction is what causes infection. It's the main target. So what was done starting off experimentally, they basically took the spike ectodomains. So the spike ectodomain means it's just extending into the extracellular matrix of various coronaviruses, and they were purified via Cephalus beads and SDS phage. And then those spike ectodomains were injected into mice. And so that basically served as the antigen that was injected into the mice. After 4 days, the spleen and lymph nodes of the mice were harvested. And so they were harvested because that's where the mouse's body was producing these B lymphocytes or B cells against antibodies or creating the antibodies. And then those B cells were hybridized with myeloma cells. And then once they were fused together, they produced hybridomas, which are basically cells that produce large amounts of monoclonal antibodies. And that's what they wanted to do in this case. And so after the antibodies were produced from the hybridomas, they were purified. And so next they wanted to identify what antibody may serve as a neutralizing factor against SARS-CoV-2. And so they wanted to assess ELISA cross-reactivity of these hybridoma supernatants that were obtained. And there was 51 of them. And so the best way that I can explain cross-reactivity, I think, is that an antibody has a specific amino acid sequence that allows it to have an affinity for its antigen. And cross-reactivity is just that that the antibody may have a high affinity for its antigen, but also may have a high affinity for another antigen. And so in this case, it could be both of the viruses that are within the same subgenus or the sarbecovirus subgenus. And so 4 of these antibodies displayed this cross-reactivity with SARS S1 and SARS S2 subunits. And then particularly one antibody, 47011, displayed cross-reactivity to both SARS S and SARS S2 infections, which are both of the S proteins that are within the sarbecovirus subgenus. So that made it an antibody of interest. And so because of that, they took complementary DNA that would encode the human monoclonal antibody 47011. And then they took that DNA and cloned it into expression plasmids in order to produce the antibody. And so the recombinant human 47011 monoclonal antibodies were produced in a human cell line. known as HEK293T. And then these antibodies were then purified from the cell culture supernatants for further use in the experimentation. And so because 47011 showed this cross-reactivity, they wanted to identify whether it binds to the cell surface spikes of SARS-CoV and SARS-CoV-2. And so they did an immunofluorescence assay. And so they took the human cell, they took human cells, HEK293T cells again, and they transfected them with plasmids that encoded the S proteins of the SARS-CoV virus, the SARS-CoV-2 virus, and another coronavirus known as MERS-S or MERS-CoV. And these viruses, the spike proteins were fused with green fluorescence protein. So when the assay was performed, the spike proteins would fluoresce a green color, as we can see in this top row here. But these cells were seated on glass slides, and then they were stained and incubated with the monoclonal antibody 47011. And when that happened, as we can see in this row, the spike proteins did not show the green color. It was actually reduced greatly, meaning that this antibody bound to the cell surface spike. And so that was what we wanted to see. So because we know that the antibody does bind to the spike protein, we wanted to know which aspect of the S protein did bind to the ACE2 receptor. So then that particular part of the S protein could be targeted. And so we knew that, it was found that there's two functional subunits that make up the S protein. There's the S1 and S2 subunits. S1 contains 4 domains: S1A, S1B, S1C, and S1D. These basically mediate cell attachment. And then the S2 subunit mediates the fusion of the viral and cellular membranes. And so here it was found that the S1B domain is what binds to the ACE2 receptor. So this wheat colored ribbon looking thing is the ACE2 receptor itself. And then here's the S1B domain. And so they were representing SARS-CoV and SARS-CoV-2 and showing that this part of the S protein is what binds to the ACE2 receptor. And then they identified that there's some slight differences as seen with the white coloration here with that particular domain between these two viruses. Okay, so because they discovered that the S1B domain is what binds to the ACE2 receptor, they wanted to just further analyze that and identify that that was certain. And so they did an ELISA assay, and these are the binding curves here. And so it was discovered that as the concentration of 47011 increases, the binding of the S protein also increases. And then in this particular assay, they were testing for the S protein, the S1A and the S1B domains, and they saw that as the concentration of 47011 increased, the S1B domain bound to the antibody, but the S1A domain was not affected at all, basically showing that the antibody had no effect on the S1A domain and was very much affecting the S1B domain. Okay, so this part, they wanted to identify if the antibodies interfered with binding the ACE2 receptors. And so first there were cells that were transfected with a plasmid that coded for the ACE2 receptor, and they were tagged with GFP. And then separately, there was S1B domain. The S1B domain of the spike protein was incubated with these various antibodies. And so when the two conditions were brought together, they wanted to test for if the ACE2 receptor was being expressed or visible um, with the antibodies there. And so it was discovered that under the 47011 condition, it was expressed the most, meaning that the S1B domain and the antibody really were bound together. And like the ACE2 receptor was not, it was neutralized more. And so the ACE2 receptor was expressed more, and meaning that the spike protein didn't bind as much, which is what we wanted. And so lastly, they wanted to measure the virus activity. And so they took pseudotyped viruses as well as the actual authentic SARS-CoV and SARS-CoV-2 viruses. And then they also took human cell line, and they transfected him with a plasmid that would code for a luciferase gene. And this would just allow us the ability to measure the infection percentage. And so what happened was the monoclonal antibodies were, as the concentration increased, okay, let me restate this. So the viruses with the antibodies were placed into cells that express the luciferase gene. And then what was measured was the infection rate. And so with the antibody present, the 47011 present, the luciferase activity was measured, and 47011 significantly decreased the infection of all the viruses, which is exactly what we wanted and is very promising therapeutically. And so overall, the results, so this antibody was identified as being able to cross-neutralize SARS-S and SARS-S2 infection. The human 47011 antibody, it does bind to the spike proteins, the full-length spike proteins of SARS-CoV and SARS-CoV-2, both within in the same subgenus. It inhibits the infection of SARS-CoV and SARS-CoV-2, the authentic viruses, in VaroE6 cells. And because it's neutralizing, it's not fully compromising the receptor and virus binding interference. And so that's the other result that they found, is that this antibody can target the S1B receptor binding domain of the virus, but does not completely compromise the spike protein and the ACE2 receptor interaction. So it is a neutralizing antibody, not fully compromising. And so I just wanted to recap everything. So the article that I just presented, they wanted to identify a monoclonal antibody that could bind the S protein of SARS-CoV-2 and neutralize the viral infection. So they took hybridomas from H2L2 mice, or they created them from the mice, and then they identified 47011 as an antibody of interest, and due to that, they expressed it in a human recombinant form. And then some of the properties that they discovered through this is that 47011 is able to bind to the cell surface spike in human cell lines. It's able to decrease the infection percentage, as we saw the luciferase activity. The expression of that was greatly decreased in SARS-CoV and SARS-CoV-2, as well as the pseudotype viruses in human cells. And this antibody targets the S1B receptor of the S protein, which is crucial because that's the aspect of the S protein that binds to the ACE2 receptor. And so the interaction with the ACE2 receptor of human mucosal cells is reduced. So overall, this is the first report of human monoclonal antibodies that can neutralize SARS-CoV-2, which is very promising. And this can be very helpful in creating antigen tests moving forward that are targeting or wanting to identify SARS-CoV-2. 47011, the properties show hope for possibly creating a therapeutic method in in altering the course of the infection of SARS-CoV-2 in a host. And it has the potential for 7011, the antibody has the potential to possibly treat or prevent COVID. And due to its cross reactivity, even other diseases that are within the sarbecovirus subgenus. And so, thank you guys so much for listening. And if you have any questions, just let me know. And these are my sources.